Welcome back to the Flagship studio here at the JP Morgan Healthcare Conference. We are rounding out day 3 with Steve Hahn, the CEO of Harbinger Health. Steve, great to see you.
You as well, Lindy. Thanks for the opportunity to be here.
Great. Let's get right into it, Steve. Cancer screening. So many good reasons to do this. We have tools today to screen for cancer in a couple of cancers. You think mammography, you think of colonoscopy, but what's the big leap that Harbinger is taking?
So we believe that, and certainly it's important that those 5 cancers that we have screening guidelines for, that it gets done, because what it has, the way it manifests itself is it identifies people with early stage disease that can be cured, and that's the key. How do you reduce the suffering and the death associated with cancer? And really one of the biggest ways is to identify it early. So we're trying to do that not just in those 5 cancers where we have screening modalities, but the ones we don't. And so by taking a simple blood test and analyzing it for circulating tumor DNA, we're able to determine a signature that allows us to identify the potential presence of a cancer. And so that drawing of blood can be turned into a screening tool for some very difficult cancers. Think pancreatic cancer, stomach cancer, liver cancer, ovarian cancer, etc. Problems and diseases where we don't have the ability to detect it early.
So we've heard, I mean, this is an incredible concept to be able to detect DNA circulating in blood. We've heard of some other products that have come to market already or are on their way that are doing something similar. What's different about Harbinger's technology?
Yes, so our technology is a biological basis in a signature. So not to get too technical, but DNA gets methylated, and it's how DNA expresses itself, and it allows for the functions of cells. And this signature we've known about for a long time in development, in the developing fetus in the placenta. And it turns out it's the same or very similar signature that can be detected in cancer. And it starts very early in the cancer evolution. So when the normal cell decides to be cancer, that gives us huge advantages. One is it allows us to detect more robustly early stage cancer. And the only thing worse than being told you have a cancer is being told that you have a stage 3 and 4 cancer. So to be able to robustly identify stage 1 and 2 like we do with mammogram and colonoscopy is the key. So that really forms the basis. We also have technology which we think will allow us to develop the tests across the entire cancer care spectrum. So screening for sure, diagnosis, and then following people with time who have cancer. And of course that's a huge problem because people get treatment, they don't often know when to stop treatment, can't detect it when it comes back, etc. So we think that this solution, because of the biology, allows us to solve problems across that spectrum.
And does it also allow you to think about screening or testing for multiple cancers in one product?
Yes.
Why is that?
So that's another advantage of this signature. This is not a lung cancer specific signature. This is a pan-cancer signature. Now, we cannot claim that it's seen in every cancer, but we know that it's seen in 20+ cancers, which is pretty good and probably represents 80+% of the cancers diagnosed around the world. So the fact that it's pan-cancer and it's seen very early allows us to look at some pretty significant cancers that cause substantial morbidity and mortality around the world.
So Steve, as you think about bringing a product to market, what does good look like in a cancer screening product?
Yeah, so we're focused on 3 things, and that really answers the question that you had. And I think it's fundamental, not just to cancer screening, but to every product along that spectrum I described. So the first is access. People need to be able to get it. As you know, lung cancer screening with a low-dose CT scan, less than 10% of the people in the United States eligible for that get that scan. And it's an access, maybe a cost issue, but access. So we believe that having this in a decentralized fashion across the world and inexpensive facilitates. So access and inexpensive test is very important to us. The second is performance. And what do I mean by performance? Well, I alluded to one, early stage sensitivity. You got to be able to pick up the cancers early because that's where you make the difference. And then the second part of it is what we call positive predictive value, or PPV. And what that means is that when you get a Harbinger test back, if it's positive, how many times does the doctor actually find a tumor when he or she looks for it? And if that number is high, say 60, 70%, that's good. If it's low, 20, 30%, that's not great. So when you think about what's good, a high positive predictive value is actually very important. Because think about being the patient, your test is positive, your doctor can't find a cancer, what am I doing next?
Yeah.
Right. And so that's the performance part of this is really important. And then the last thing is what we call clinical informativeness. What are we telling the doctor might be the best next step? And of course, that's a sweet spot for me because I care so much about that interaction between patients and doctors. And so we have something called tissue of origin, which allows us to tell the doctor, this is where you need to look to find the cancer. Critical part of this.
So you talked about 3 really important characteristics of a good product. It has to be, the performance has to be there, access has to be there, and then what does the doctor do with that information?
Absolutely.
So as you are thinking about the technology and the performance pieces and the data that you have in hand right now, where are you seeing the commercial entry point for Harbinger?
So this is the great thing about a flagship company is that we have a platform and the biology forms the basis of our platform with the technology. And so now we can think of across an entire cancer care continuum, where do we intervene? So we've decided that one of the best ways to approach this is to look at what is the underlying risk of someone for cancer. So it could be you and me where the risk is, low because we don't have symptoms and maybe not other risk factors. It could be someone with genetic abnormalities like BRCA1 and 2 where the risk is higher. Smokers would be another high risk group where it is. Obesity has some risks associated with cancer. And then of course people who present with non-specific symptoms where cancer is higher on the differential diagnosis. So we think that we would like to have products first along that risk spectrum because the need is great. The ability to detect a cancer is great, we believe, and also we think the pathway to commercialization is shorter and we can get to patients more quickly.
What kind of timeline are we talking about? When can we expect to see Harbinger rolling out first products?
So we're looking to have what's called CLIA. It's the type of laboratory that we run that's regulated by the US government. We have a CLIA laboratory and we'll have analytical and clinical validation of our first products this upcoming year.
Very exciting.
Yes.
And for you as CEO, where's your focus this year? Is it all on that commercial product? Are you thinking 2 steps ahead?
We're thinking across across all of that. So how do we make our performance better? We think we have excellent performance that'll fit a number of different products, but where do we need to work further in the R&D side to make it better? Because we don't want to stand still. We also are working on the commercial side of things and the laboratory side of things. But we're also looking at reaching out and finding the best places to perform these studies. So we're thinking about international expansion as well. Because a very interesting part of this is that the underlying genetics of a person depends upon their background, race and ethnicity, and where they live. And we have found in our studies that even within the United States, that factors into what the sort of baseline DNA looks like. If we don't have enough diversity scientifically and clinically in our samples, we can't build our AI ML algorithms to get the best answer for everybody. And we want to make sure this is worldwide acceptance across all groups, certainly in the US, but around the world.
So, Steve, it's tremendously exciting to see an early idea take shape, and now we're starting to think about how we can bring that solution to patients. Thanks for being here today.
Thank you, Lindy. Really terrific to be here.